So hi everybody and welcome to Taxonomy and Phylogenetics for Conservation. So we're going to be looking at taxonomy and phylogenetics purely just kind of skimming the surface, as it were, of these subjects, enough for us to be able to successfully complete our practical investigation and then our subsequent quiz. It is a cut down version of the lecture from the evolution and phylogenetics module. If you do feel like you want a little bit more detail, do feel free to register for the evolution module and take a look. If you don't feel like you're still stuck, then there are loads and loads of resources on the internet for you to use. You can also look in books. If you're still stuck and you do require more help, do feel free to post on the forum. So this presentation aims to give you an introduction to taxonomy and phylogenetics that will be relevant for the conservation module. Obviously in the conservation module what we're taking a look at is the clouded leopard and whether or not it, because of them being in different areas of the world and cut off from each other, whether or not gene flow was halted and they actually developed into two separate species, or whether or not they're not different enough to be two separate species and they should remain as subspecies. But to understand that, we first need to understand how we actually classify organisms. So how do we classify living things? So one of the oldest sciences that is devoted to understanding organisms' relationships and their evolutionary history is called taxonomy. So taxonomy is the science of classifying living organisms into groups, and this is based on the binomial system that was first proposed by Carl Linnaeus in 1735, and that's that guy on the left. So binomial in biology means two-part names. So for example, Homo sapien, Homo neanderthalensis, Pan troglodytes, I'm sure you can think of a few others. And they consist of the genus and the species. So Homo is our genus and species is sapien. In taxonomy, organisms, they are also called taxa, are grouped according to shared characteristics or traits. Organisms are classified and ordered into 8 descending categories, and each descent includes less organisms than the one before it. Now what I mean is, I'm not saying that there are more domains than there are species, because these are your 8 classifications over here. That's the reverse is actually true. What I'm saying is the organisms share less and less organisms as we work our way down. So for example, there are 3 domains of life. There is archaea, eukaryota and prokaryota. So we belong to domain eukaryota. Now all of life fits into those 3 domains. So as you can imagine, we share domain eukaryota with hundreds of thousands, if not millions, of different organisms. As we work our way down to the next group, we are kingdom Animalia, which means we're animals. Now, as you can imagine, we don't share kingdom Animalia with as many organisms as we did domain eukaryota, because in fact eukaryota also breaks off into fungi and plants as well. So we don't share that kingdom with them as well, so we're sharing less organisms. And as we work our way down, we share it less and less. It becomes more and more specific. The classification becomes more specific to the species. So as we work our way down to genus and species, we are in fact the only living members of our genus and our species. So what I'm saying is, as we work our way down the groups, we share that category with less and less organisms. Now what I haven't included is subspecies. Now I haven't included that because it's really, really difficult to define. There is, like defining species, which is also very difficult, defining subspecies is even more difficult, and there's lots of debate over whether or not species are subspecies or whether they get that classification or not. That's really difficult to go into. If you want to take a look at how we classify subspecies, do feel free, and equally if you want to take a look at how we classify species, then that's also a really great thing to take a look at, because as I'm sure you're already aware, defining species is difficult enough, let alone subspecies. But it is something that we come across for conservation, like for example in our clouded leopard. So in our investigation, what we want to know is whether the clouded leopard is in fact 2 species, gene flow was halted for long enough because they were separated geographically, and whether or not they became different species, or whether or not the differences aren't great enough and we should just regard them as subspecies. So I just put the full classification of the clouded leopard up, and I also put the human one up so you can compare. So we share all of the classifications up until class, so we are both mammals, and then obviously we start to split off, we are primates, they are Felidae, and we work our way down that way. So Neophelis nebulosa is our clouded leopard, and Homo sapien is obviously ourselves. So that's just an example of a classification of our clouded leopard. Now, but how do we get to this classification? So we know how it's classified, but how do we actually go about classifying the organisms? So one way to classify living organisms is to construct a phylogram or cladogram, and these are commonly known as phylogenetic trees. So Charles Darwin drew one of the first phylogenetic trees, but he did not draw the first phylogenetic tree. It's probably important I mention that. So a phylogenetic tree is a tool used by evolutionary biologists to visualise organisms' relationships and their evolutionary history. But obviously back in the day of Charles Darwin and Carl Linnaeus, DNA technology obviously didn't exist. So many original phylogenetic trees used shared physiological traits, such as whether the organism had wings, flippers, or legs, or whether the organism had similar structures in their anatomy, so for example, similar eye structure. But can you see a problem with that? So if we take a look at the organisms on the screen at the moment, all of them have wings, but do you think they're closely related? And I really hope you're saying no, because on here we have, in fact, an insect, a bird, and a mammal. So they're not very closely related at all, but they all have wings. And this is because of convergent evolution. So many organisms living in similar environments have evolved similar structures. So for example, thick fur for cold environments, wings for life in the air, flippers for life underwater, and legs for life on land. However, this does not mean that they are closely related. Similar traits, structures, or behaviours can evolve independently many times in distantly related lineages. So for example, wings that evolved independently in birds, mammals, and insects. Similar environments instill similar selection pressures on organisms, which drives the independent evolution of these traits, structures, or behaviours. So instead of using shared physiological traits and similar physiological structures, we now use genetic sequence. So sequencing genetic code has become far less expensive and far more readily available, which means more organisms are having their entire genome sequenced, which is great for people like me who kind of specialise in DNA sequence and we like to analyse it, because it means far more DNA sequence to compare. We base phylogenies using genetic code on the basis that organisms that are closely related will have more identical or similar genetic sequence with each other than organisms that are distantly related to them. And I'm just going to show you that in a diagram. Okay, so this might look a little bit complicated to you, and I don't want you to kind of freak out. So here in front of us we have 3 made-up genetic sequences, and we've got 3 taxa. I'm calling them taxa 1, 2, and 3. And all I've done is I'm doing how phylogenetic programs work. So I've lined them up and I'm looking for differences. So 1 and 3 are identical at 25 out of 28 nucleotide positions. 1 and 2 are identical at 21 out of 28 nucleotide positions, and 2 and 3 are identical at 21 out of 28 nucleotide positions. So that means that 1 and 3 are more closely related than 1 and 2 and 3 and 2 are. I know this can be a little bit tricky to get your head around, so if you are stuck, please do feel free to post on the forum, and I will of course do another tutorial if that's what you need. Just let me know, because I know this can sometimes be a little bit daunting, but it's just based on similarities and differences. All the genetic program is looking for is who is more closely to who, based on the theory that organisms with more genetic code are more closely related than others with less genetic code. Okay, so now we know how they build a phylogenetic tree. I'm sure you've all seen one before. What does it actually show you? So I've kind of done it in one block picture here. So I'm just going to start off with time. So a phylogenetic tree represents time, and time is going in this direction. It's often in millions of years because the evolutionary differences, the variations, take millions of years to happen and millions of years to evolve. So we're looking on the scale of millions of years. So we'll start here. So these are your present day species. So for instance, I'm really interested in species A to H, and I want to know how they're all related. And that's what this phylogenetic tree is showing me. It is showing me how species A to H are related, and it's also constructing their evolutionary history. And I will go into that in just a second. Now the one that you might see sticking out here is your root species, and that is because we choose a species that is not closely related to our other species in the phylogenetic tree. We want it to stick out. We don't want it to form in the tree, and we want to make it a root. Now the reason we want a root species is because it helps us reconstruct the evolutionary history. It helps the program do that. So when we select our root species, and we've chosen for our root species in our practical, we've chosen the domestic cat, and that's because although it's kind of closely related to the clouded leopard and the other species in the phylogenetic trees, it's not too closely related. It won't fit in part of the tree and start to integrate itself in these species here. It'll stick out at the bottom, which is just what we want. It's what the program needs to reconstruct the evolutionary history. And it uses this root species to reconstruct these here. Now this is telling you about the evolutionary history of the species. So this is your common ancestral speciation events. So we can kind of trace it back. So for instance, this here is representing the common ancestor of species A and B. And this here is representing the common ancestor of species C and D. And this here is representing the common ancestor of all species A, B, C, and D. And this here is representing the common ancestor of species A, B, C, and D as well. And obviously this one is also representing the common ancestor of all of them, So this one here is representing the common ancestor of species A to H, because as you can see it then branches off down here, and this common ancestor is representing only the common ancestor of species A, E, F, G, and H. And then again, this is the common ancestor of species E to F, and this is the common ancestor of G to H. Okay, so this is just telling you about speciation events, and a speciation event is when one species splits into two. But I will be going into that into a little bit more detail. I just want to show you a different phylogenetic tree, so please don't panic, the information shown here is the same. Phylogenetic trees can actually be drawn in a number of ways. We won't be using one like this, ours will look like this, but if you ever do come across one that looks slightly different, don't panic, it's showing you exactly the same thing. So these are your tips or your external nodes, these are your internal nodes, and this is your root, so E is your root. Okay? So I'm just going to go into outgroup species a little bit more. So we use outgroup species to root a phylogenetic tree. So an outgroup species is distantly related to the other species in the phylogenetic tree. So I say a good outgroup species is in the Goldilocks zone. You don't want to use a species that is completely distantly related. I mean, you could, but it's not as useful as using a species that is slightly more closely related. So for example, in ours we chose the domestic cat, but because it's not too distantly related, it's not so distantly related, so it's kind of like it's a nice little Goldilocks zone in between. So by having an outgroup, the phylogenetic software can reconstruct the evolutionary history of our species, forming the internal nodes. You may come across other people using the term root species instead of outgroup. That's absolutely fine. You can use root species, you can use outgroup, they mean exactly the same thing. Root species might be easier for you to remember because it does form a root at the bottom, or it might be easier for you to use outgroup species, that's absolutely fine. It's also important for me to mention that the root species, although it tends to be at the bottom, sometimes the phylogenetic tree can, the software, can put it at the top. And that's a little bit counterintuitive to root species, which is why some people use the term outgroup as well. That's absolutely fine, as long as it sticks out and it's forming clearly its own little node over here, that's absolutely fine. So I've highlighted here your external nodes. So the external nodes tell you where the present day, so extant species are and their relationships between one another. And internal nodes are in red. And internal nodes tell you how the present day species got to where they are today by showing you the speciation events. And again, that's your common ancestor and speciation events there, in case that's easier for you to understand using that diagram rather than that diagram. So speciation events are when one species splits into two or more, and it is the driving force behind Earth's astonishing biodiversity. Now I want you to pause this video and watch the educational video by Cornell University on speciation. It's fantastic. It takes what is really complicated for me to describe to you just via kind of images and talking alone, and it doesn't a video, and I find it really, really great. So if you take a look at the speciation video by Cornell University, it should be at the bottom of this page, just below this presentation. If it's not for whatever reason, then it's also on the evolution lecture page as well, so you can find it. I'd pause this video and watch that before we continue. Okay, so moving on to a few more key terms. So sister taxa refer to the closest relative of the taxon on a phylogenetic tree. So if you remember that kind of basic phylogenetic tree we made earlier, 1 and 3 would be sister taxa. So we just call that sister taxa right there. Okay? So 1 and 3 are sister taxon. So again, common ancestors, I talk about them a lot. So A and B, this is the common ancestor of A and B, and this is the common ancestor of A, B, and C. Ancestral lineages. So an ancestral lineage is like taking a journey along the phylogenetic tree. So I'll work our way along this one. So this one here is representing the ancestral lineage of A, B, and C. This one here is representing the ancestral lineage of C, because as you can see, there is nothing else on here. There is only C, so that is ancestral lineage of C only. This is the ancestral lineage of A and B. This is the ancestral lineage of B only, and this is for A only. Okay? We won't be using these key terms at all in our practical, but I just think it's important for you to kind of know these terms exist in case you come across it. If you ever do your own research on the internet and you come across these terms before, don't worry. Although a lot of phylogenetic terms are very easily found if you just Google them or look them up. So hopefully that's everything you need to know. So this presentation should have gone into enough detail for you to be able to complete the practical and final quiz. If you're stuck at any point, go through this presentation again, use any of the extra resources. There's a really great one called Understanding Evolution, which is linked in the extra resource section. It's absolutely fantastic. I used it throughout my degree, and I know that my professors and my tutors also use it as well. It's brilliant. But if you're stuck at anything, they explain it wonderfully. Look up other extra resources on the web or post on the forum. If you're finding this presentation difficult to understand, I need to explain things better, do tell me, and I will post a quick tutorial for everyone to see. I will go through points in detail that people need to understand better. People tell me that I didn't explain it properly, that's perfectly okay. Just let me know and I will do what I can. So well done guys, and best of luck for the practical.